Hello and welcome. Let's review Chapter 9 here today with information in regards to muscles and muscle tissue. So first thing here, let's get this presentation enlarged. We've got slideshow. In action. All right. So here now, when we go through and we talk muscles, I'd like to cover a little overview of muscle tissue with you guys here. First, I'd like you guys to know muscle tissue makes up nearly half of the body's mass. We'll see when we talk muscle tissue makes up nearly half of the body's mass. Muscles are going to be special because of their ability to transform. They're going to be important because of their ability to transform chemical energy. They have the ability to transform chemical energy, ATP, into directed or mechanical energy, as we can see here. So they have the ability to transform chemical energy, ATP, adenosine triphosphate, right, as we've seen, to directed mechanical energy. And the mechanical energy allows us to then exert force. So allows a task to be completed, we could say there, by the transformation of chemical energy to mechanical energy. So that's one, again, special characteristic we can see in relation to muscle tissue. Now, when we go through and we talk muscle tissue, I want to discuss with you the different types of muscle tissue. Now, when we go through and we talk different types of muscle tissue, here we're going to see we have three different types of muscle tissue. We have skeletal muscle. We are going to go through, we're going to check that out in full detail. We have cardiac muscle, we'll do the same thing. And then we have smooth muscle. Now, when we go through and we talk first muscle tissue, I'd like you to know of the term muscle fiber. You'll see this term is going to be a name that's going to be given to skeletal and smooth muscle cells because they're going to be elongated cells. So here when we go through and we talk muscle fibers, now you've got to understand what he's talking about is he's talking about a muscle cell. So here when we go through, let's talk skeletal muscle tissue first. We'll go through and we'll look at in full on detail. Now here there's some prefixes you can appreciate here at the bottom of the screen as well, all in reference to muscle. So you can understand those there as well. Then when we talk skeletal muscle, so here we'll go through these different components in relation to skeletal muscle, and we'll check out same thing with cardiac and smooth muscle, and we'll look at these images basically depicting all three. So let's just move right to these pictures here and this slide. Now here when we talk skeletal muscle tissue is what we're going to look at now in greater detail. So skeletal muscle tissue is going to make up skeletal muscle. Now when we talk skeletal muscle is going to be found as an organ that's attached to, so it's an organ that's going to be found attached to and covering our bony skeleton. So it's found attached to and covering our bony skeleton. Skeletal muscle fibers. Now when we look at one skeletal muscle fibers are the longest muscle cells that we're going to go through and we're going to see. These skeletal muscle fibers, they have these obvious stripes that you can see, or striations you can see there in relation to them. So you can see that here in this first image, you've got all these obvious striations. And here when you're looking at just one skeletal muscle fiber, you can appreciate those striations, those stripes there again. So here when we go through and we talk skeletal muscle, excuse me, I want you guys to know skeletal muscle is going to be voluntary. It's under voluntary control. It's going to be under voluntary conscious control. And skeletal muscle is going to be responsible for the body's overall body mobility. It will be responsible for the body's overall body mobility. Right? Locomotion, moving from one place to another, or manipulation of the environment, right? I don't want this here, I don't want that here, let me move this from here, I'll put this here, let me move, these papers from here over to there. So manipulation of the environment, you can see there as well. Now when we go through and we talk cardiac muscle then, so when we talk cardiac muscle, you can see cardiac muscle tissue is only going to be found occurring in the heart, so it occurs only in the heart. And when we talk cardiac muscle is also going to be striated. It is also striated. You can see it's got obvious striations Here we can see that in this picture. You can see this here in the simple image on the bottom where it's depicting one muscle cell. You can see those striations there also. Now, when we talk cardiac muscle tissue, so only occurs in the heart, striated, and also it is going to be under involuntary control. It's under involuntary control, so it's not subject to conscious control. You are not going to consciously control your heart. Involuntary, you have no conscious control over how your heart is going to beat. Right, unless you obviously go from parasympathetic to sympathetic and, vice versa. So cardiac muscle cells, I want you to know cardiac muscle cells are going to contract. They contract at a steady rate. They contract at a steady rate. And the steady rate that these cells are going to contract at is going to be set by the heart's pacemaker cells. It will be set by the heart's pacemaker cells. And then, like we said, we'll see neural controls are going to allow the heart to speed up for brief periods. Neural and we'll see even certain stress hormones can get involved there as well and allow this to kind of beat faster there as well. So when we talk neural and hormonal controls, can allow the heart to speed up for brief periods there as well. Next, then we've got smooth muscle. So when we talk smooth muscle is going to be found in the walls of our hollow visceral organs. It will be found in the walls of our hollow visceral organs, such as the stomach, the bladder, urinary bladder, we can say there, the respiratory passages, reproductive passages there also. Now its function is going to be to force fluids and other substances. So it's responsible for forcing fluids and other substances through these internal body passageways. Through these body passageways. We also refer to smooth muscle as visceral muscle because of, again, where it's found. It is non-striated, hence the name smooth muscle. So non-striated. And also under involuntary control. Right, we have you'll see we're not going to be able to control its activity. So again, it's not subject to conscious control. So under involuntary control. Next, then let's talk some special characteristics in relation to muscle. So here's some more detailed images. We're going to go through all of this, and we're going to look at all of this in full-on detail. And here we're going to be able to see now when we go through and we talk skeletal muscle, cardiac muscle, and smooth muscle, you can see some more differences in between each of them as well. Now here we can see when we talk skeletal muscle, we've got what we call neuromuscular junctions that are going to be found there between one muscle fiber and one nervous tissue basically component there. So here you can see we have neuromuscular junctions, which we're not going to find then when it comes to cardiac muscle. And also, it's not unitary muscle, it's a multi-unit muscle when it comes to smooth muscle, we're going to see there. So it's going to be a little bit different there when we talk basically neuromuscular junctions. Gap junctions then, because of the way these fibers are being innervated, we don't have any gap junctions here in skeletal muscle, but we do have them in both cardiac and smooth muscle. So one cell gets excited, it's going to excite the next cell, the next cell, the next cell, the next cell. So they'll be in this coordinated fashion there. So let's talk some special characteristics of muscle tissue. Now when we talk special characteristics of muscle tissue, here we can see some special characteristics we've got here. First special characteristic that I'd like you to know about is going to be this characteristic known as excitability. Excitability. Now, when we talk excitability, this is basically talking about how muscle is responsive. So it's talking about its responsiveness or its irritability, you can say. Now, when we talk excitability, here we're talking about how muscle tissue is able to receive and respond to stimuli. It is able to receive, so it has the ability to receive stimuli and respond to stimuli. Right? We didn't talk about this type of stuff in relation to epithelial tissue. We didn't talk about these types of characteristics in relation to connective tissue. So these are special characteristics, again, in relation to muscle tissue. Next is contractility. Contractility, when we talk contractility, muscle has the ability to shorten forcibly. It has the ability to shorten forcibly when adequately stimulated. So it can shorten forcibly if we adequately stimulate it. Third characteristic we're going to see here, a special characteristic, is going to be extensibility. Extensibility. Now, when we talk extensibility, here we're talking about how it has the ability to be stretched or extended. So muscle has the ability to be stretched or extended. Fourth, then we have elasticity. Elasticity. Here, now when we talk elasticity, we're talking how muscle has the ability to recoil. It has the ability to recoil. Now, when I say recoil, that means it has the ability to go back to its resting length after having been stretched. So it's elastic, you can think of, right? Like around our sometimes pants and shorts and whatnot. There, you have elastic, and basically it's saying you have the ability to stretch them out, and then they go back to the resting length. So the same thing here. They have the ability to recoil to the resting length after having been stretched. So 4, then number 5 I'd like you to add in there is conductivity. Number 5 is conductivity. Now, when we talk conductivity, here we're talking about how muscle tissue has the ability to conduct electrical impulses. You guys learned a lot about action potentials in the last exam. You guys talked about that there. We're going to see them here again. So conductivity is the 5th characteristic. Has the ability to conduct electrical impulses, to conduct action potentials, nerve impulses. Let's talk muscle functions. Now, when we talk muscle functions, I'd like you guys to know muscles are going to perform 4 important functions for the body, and then we'll look at some additional functions there as well. Now, when we talk 4 main functions are going to include, number 1, producing movement. Muscle tissue is going to be responsible for producing movement. Now, when we talk muscle tissue, it's more specific to skeletal muscle, as you can see there. So when we talk movement, here we're going to see, okay, first, in regards to skeletal muscle. Then we're going to see movement in regards to cardiac muscle. Then we'll see movement in regards to smooth muscle there as well. So we talk movement first, movement of bones. We talk movement of bones, right? Locomotion, manipulation of environment, we said there. Skeletal muscle. Cardiac muscle, we talk movement. Cardiac muscle is going to be responsible for moving the heart, and when the heart moves blood. And then smooth muscle. Smooth muscle is responsible for moving or coursing digestive, urinary, and reproductive components. Digestive, urinary, and reproductive components. Because we talked about where that type of muscle tissue is going to be found. So producing movement, first function. Bam. Next, then we've got maintaining posture and body position. Maintaining posture and body position. Now, here mainly in relation to skeletal muscle, right? Skeletal muscle is constantly making tiny adjustments. You've got contractions and relaxations happening now of these different muscles, all to counteract gravity to keep us erect. All to counteract gravity and to keep us erect. So maintaining posture and body positions. Stabilizing our joints, again, coming back to skeletal muscle. So when we talk stabilizing joints, joints are going to get stabilized because if you recall when we talked about joints in anatomy, we said joints are going to be the weakest part of the skeleton. Nonetheless, they're able to resist various forces, right? So that's going to be even better when you have the muscles actually in a contracted state. So here when the muscles are in a contracted state, it's going to be, you'll see, harder to destabilize these joints. So stabilizing joints, you can see another function. So joints get stabilized as these muscles pull on bones and cause movement. Next thing we've got generating heat, heat generation. Now we talk generating heat, heat generation when muscle contracts, especially, right, skeletal muscle. So muscles generate heat as they contract. They will generate heat as they contract. Now additional functions like we mentioned. Now we talk additional functions are going to include, number one, we've got skeletal muscle. Now we talk skeletal muscle, you'll see is going to be responsible for protecting our internal organs, right? We've got our 8-packs, the 8-pack muscles there, along with the rest of the obliques, help to protect our organs. We have no bone protection there, so just muscles. Same thing with our thoracic organs, right? Between the ribs, we've got basically space, intercostal space, and then that space is occupied by muscle. And the muscle there, just like with the abdominal muscles, again, helping to strengthen it, not only just by their presence, but also by the way they come in, right? They come in at 90-degree angles. You should have learned in anatomy, and those 90-degree angles all help to make basically these V's, and that's helping to protect and give more strength to that area there. So protecting organs, you can see there. Next one we've got is smooth muscle. Smooth muscle is going to form valves. So it forms valves. And when you talk valves, just like in plumbing, valves are going to help to regulate the passage of flow. Smooth muscle is responsible for constricting pupil size. So it dilates and constricts the pupils. And then also forms the erector pili or piloerector muscle, right? That's the muscle that's attached to the hair follicles, and it's responsible for causing goosebumps. You learned about in anatomy as well. So again, now all functions in relation to muscle tissue. All functions in relation to muscle tissue. Next then, let's talk skeletal muscle. Let's talk in greater detail, further detail of skeletal muscle. Now when we go through, we talk skeletal muscle. Skeletal muscle, first thing, let's talk gross anatomy of skeletal muscle. And we talk gross anatomy of skeletal muscle. Let's talk why skeletal muscle is considered to be an organ. Now, when we consider skeletal muscle to be an organ, going back to the definition of an organ is a structure that's going to be made up of 2 or more primary tissue types, right? More commonly 4. So here when we say it's an organ, first thing we can take right off the bat is muscle tissue, right? Muscle tissue. And we're going to go through and we're going to see now the rest of these components there and how they help basically muscle to become an organ. So here you can see each muscle is going to be served by 1 artery, 1 nerve, and 1 or more veins. So we'll go through and we'll see blood vessels, nerve fibers, and connective tissue are also going to be present here. So when we talk nerves and blood supply, first let's talk nerve. I want you guys to know each muscle is served by 1 nerve, 1 artery, and by 1 or more veins. Okay? The reason why 1 or more veins, we want to make sure we can pump out all of this waste that gets accumulated or that gets produced there so it doesn't start to accumulate from the muscle actually doing activity. Now each skeletal muscle fiber is going to be, innervated with a nerve ending that controls its activity. So each skeletal muscle fiber is innervated with a nerve ending that controls its activity. Now, you'll see this nerve is going to enter and it's going to exit basically a central part. And then it's going to branch through all the different connective tissue sheets. And again, like we said, each skeletal muscle then is going to be innervated with a nerve ending. So you can see that here also. Each skeletal muscle fiber is going to be supplied by nerve endings that control its activities. So along with those arteries, you can see these blood vessels as well making their way in. Now when we talk blood vessels, skeletal muscle has a rich blood supply. Skeletal muscle will have a rich blood supply. So has a huge nutrient and oxygen demand. Has a huge nutrient and oxygen demand, especially during periods of activity. So since it's using lots of nutrients and oxygen, it's going to be generating large amounts of waste as well. So there you can see why then, in some cases, one or more veins are going to be found there. So connective tissue. When we talk connective tissue in relation to our muscles, so we got muscle tissue, connective tissue next. Then that's two primary tissue types, so bam, we've hit the criteria, but we'll get the other two there as well. So we talk connective tissue. Connective tissue is going to wrap and hold together individual muscle fibers and also bunches of muscle fibers as well. So first definition I want you to know about in relation to the connective tissue is going to be in relation to epimysium. So again, these are going to be connective tissue sheets that are going to be wrapping and protecting skeletal muscle. So they support the cells and then they reinforce the whole muscle there as well. Now going from external to internal is how they're going, but we're going to go from, you can see here, epimysium. Okay, so we're going to do the same thing. We're going to go from external to internal. So let's start epimysium. Epimysium is the coat, you can think, or the overcoat is the better term, like a jacket you can think of. It's the overcoat. So we talk epimysium, it's the coat or the wrapping of dense, irregular connective tissue. Dense, irregular connective tissue that's going to be found surrounding the muscle as a whole. It's found surrounding the entire muscle. And it's may blend with the fascia, with the superficial fascia there as well. It itself is also considered fascia. Here you can appreciate that epimysium. Now here we've got that epimysium. You can see surrounding the whole muscle. So it's like an overcoat. A layer of dense, irregular connective tissue. Dense, irregular connective tissue. You should have learned the differences between dense, irregular, and regular, right, throughout your career. And that's why now you can, make sense of that. It's going to be found surrounding the whole muscle again. Be found surrounding the whole muscle. Next one we have perimysium. So we break this muscle down further, right? We're not going to get into tendons and all that. That's all anatomy. You've gone through and you've talked about how the epimysium extends beyond this muscle as a rope-like tendon to come into contact with then the bone there, giving rise then to what we refer to as a indirect type of attachment versus a direct attachment and all of that. So here then you can see now when we break this muscle down, we break the muscle down into a whole bunch of different fascicles. So a whole bunch of fascicles. Now these fascicles, we're going to break down one of them. We look at one of them. Here you can see in relation to one fascicle, you've got then perimysium. So when we talk perimysium, perimysium is going to be a coat of fibrous connective tissue. It is a coat of fibrous connective tissue that surrounds each individual fascicle. That will be found surrounding each individual fascicle. So a coat of fibrous connective tissue that surrounds each fascicle. Now we break these fascicles down even further. We break them down even further then into a whole bunch of muscle fibers. So we talk muscle fibers now. We've got, again, connective tissue in relation to the muscle fiber as well. Now the connective tissue we're going to find here is going to be referred to as endomysium. Endomysium. Now endomysium is going to be areolar connective tissue. If you recall, I don't know, you guys who have had me since 107, you know I refer to areolar as spaghetti and meatballs. So if you go back and you think about that one, that's what we're talking about there. So areolar connective tissue that surrounds each individual muscle fiber. And then internal to that, you're going to find the muscle's plasma membrane, and we'll talk about that here in a few seconds also. Do we have a picture of that? I believe so. So here you got the fascicle. We'll break the fascicle down. You can see from the whole muscle. And then here we've got the fascicle in greater detail. The fascicle then gets broken down into individual muscle fibers. Now those individual muscle fibers we can appreciate here when we zoom in and look at that individual muscle fiber in greater detail, you can appreciate how you've got that endomycelium here on the outside. So again, areolar connective tissue, and internal to that, you've got the sarcolemma, which we're going to talk about here in a second in greater detail. And then here you can see we removed basically everything from there. We've pulled basically just one myofibril out because that's the one thing we need to pull out of the cell and really look at. We don't care about the mitochondria now. We've already done that. You guys know everything about that. The nucleus, you've already done that. You know everything about that. We're not going to be looking at the ribosomes, the endoplasmic reticulum, right? That's all been done. You know all that. This is the one thing that you don't know and you haven't looked at in relation to these cells here in physio, because in anatomy you've done it, and in 107 I'm sure you've checked it out there as well. So third time then is the charm, right? So here when we go through, then we talk, we're talking now microscopic anatomy is what we're looking at here. So this is the microscopic anatomy of a skeletal muscle fiber. So the microscopic anatomy of a skeletal muscle fiber. Now I want you guys to know each skeletal muscle fiber is, again, a long cylindrical cell. It's a long cylindrical cell with multiple oval nuclei. So these are multinucleate cells. And they're going to be covered by a sarcolemma. Sarcolemma is going to be the plasma membrane. It's a cute or fancy term you can think of that we use here in relation to muscles' plasma membrane. So here we don't refer to it as just the plasma membrane. Now we're going to refer to it specifically as the sarcolemma. So sarcolemma is the plasma membrane of each multinucleate skeletal muscle fiber. Now, we have also here sarcoplasm. Sarcoplasm. Now, when we talk sarcoplasm, sarcoplasm is the cytoplasm of a muscle cell. It is the cytoplasm of a muscle cell. Now, when we talk sarcoplasm, similar to the cytoplasm of other cells, but here you'll see it contains unusually large amounts of glycosomes. It contains unusually large quantities of glycosomes, and also you saw another difference is myofibrils, right? We didn't see that in any other cells, or even in a typical cell when we talked about. So these cells, their cytoplasm is unusual because it contains an unusually large amount of glycosomes and myoglobin. So you may ask, what is glycosomes and what is myoglobin? In anatomy, I'm sure you've gone through them there as well. But here, I'll tell you guys also, glycosomes are going to be granules of stored glycogen. They are granules of stored glycogen. And then what they do is they provide glucose. They provide glucose during periods of muscle activity. And we talked in here as well, right? We said glucose is going to be found stored in the body in a couple of different ways. Glycogen was going to be one of them, and then we said in a couple of areas that's going to be found is going to be muscle, and you can see the liver. So glucose also gets stored in adipose as well, right? I mean, if it's been picked up by these other two tissues and we still have glucose floating around, then adipose is going to start to pick it up. And we've got to get it out of the blood. Now we talk myoglobin. Myoglobin is going to be a red pigment. It is a red pigment that stores oxygen. So it's a red pigment that stores oxygen. And it's similar to hemoglobin. So hence the name myoglobin, hemoglobin. Similar to hemoglobin. Stores, red pigment that stores oxygen in muscle. All right, mitochondria you know about, nucleus you know about. Okay, myofibrils. Let's grab myofibrils. I'm sure some of you guys know about myofibrils as well. Let's grab myofibrils, and we talk myofibrils. Myofibrils, you can see now in this picture on the bottom, myofibrils are going to be rod-like structures. They are rod-like structures, and they're found in numerous quantities. They are found in numerous quantities in each muscle fiber. So rod-like structures found in numerous quantities in each muscle fiber. They make up about 80%. You can see they make up about 80% of a cell's volume. And again, I said you can see that by looking at the picture above. You can see how many myofibrils are going to be found in relation to the muscle. You got mitochondria in there also. We can see in the nucleus in there also. But again, these myofibrils are taking up an abundant amount of area in there. So they make up about 80% of the cell's volume. Now, these myofibrils are important because they're going to contain the contractile elements. They're important because they will contain the contractile elements, what we refer to as the sarcomeres. Okay, they're going to have the sarcomeres. We talk sarcomeres, we're going to see are going to be the contractile units. So they're going to contain the contractile elements, which we refer to as the sarcomeres. So we're going to see, get back into here. Now, when we talk myofibrils, okay, so they're going to be found in numerous quantities. Now, they contain the actual contractile elements, the sarcomeres, as we said, which are going to contain even smaller rod-like structures that we're going to look at, which are going to be referred to as the myofilaments. So we'll look at those myofilaments and in full-on detail. So let's go through and let's see these striations. Now, when we look at these striations here are a repeating series of dark and light bands now that are going to be found in relation to the myofibrils. So when we look at one of the myofibrils in greater detail, here you can appreciate that myofibril, but let's look at that in even better detail. So to do that, here we can see this picture there. Okay, so this is much better. So here we went through, we talked, you can see now, when we magnify the sarcomere, what we're going to see in greater detail then is going to be this image right inside of here. So this is the sarcomere. These are all the components. We're going to go through each and every one of these components, and we're going to talk about every one of them, and we're going to see how they're going to be related in relation to the contraction of muscle. So right, that's what you're here to learn, is that physiology, how this muscle is going to contract. So here then you can see in even greater detail, the two major components, the thick and the thin filaments, then is what we're going to really take everything down to and look at them. And that's what we're seeing right inside of here. So let's do this. Let's move over to, again, this slide, and you can appreciate all the components we've gone through and we've talked about here. Now we're going to look at some modified structures that we're going to find only in relation to skeletal muscle, and they're going to be the myofibrils, the sarcoplasmic reticulum, and the T-tubules. We're going to see the same structures in relation to cardiac muscle as well, but they'll be a little bit different. And then obviously not in smooth muscle, because smooth muscle is smooth. It doesn't have striations. Those striations is what we're looking at, right? So it doesn't have these structures here. It's going to have other components it's going to use for contraction. And we'll look at that when we get there. Now, we talked myofibrils. We talked about everything we see here as well. So here then you can actually appreciate two muscle fibers, or like you can say a very small amount of one, probably like an eighth maybe of one. You can see right inside of here, and then like a whole one right inside of here, another whole one right inside of here, and then probably like a like a 16th maybe there, and probably like a an eighth of one right down in here. Probably not even a full quarter even yet inside of here. So that's kind of what you're seeing here. So we're going to look at one muscle fiber. You can see right inside of there. You can orient yourself with the nuclei there. And then here we've got a whole bunch of striations. So again, repeating series of dark and light bands, right? That's what you're seeing. Dark band, light band, dark band, light band, dark band, light band, dark band, light band. So dark band you can also refer to as the A band. Light band we can also refer to as the I band. So you can say repeating series of I bands, A bands, I bands, A bands, I bands, A bands, A bands, I bands, I bands, A bands, or whatever you want to say there. Let's look at those then in full-on detail. Here's one myofibril then that we've extracted away from the rest of the myofibrils. Okay, you can see how detailed that picture is. Very nice picture. You can see the mitochondria there then in full-on detail. Then we can appreciate even the nuclei there in full-on detail as well. Now those striations, so when we look at these striations in full-on detail, this is what we're going to get into. We're going to get into the H-zone, the M-line, Z-disc, the thick and thin filaments, and basically that's what's making up the whole sarcomere. So we'll look at all of that in full-on detail. Now here when we go through and we talk A bands, I bands, dark bands, light bands, that's what we're going to go through. We're going to orient everything in relation to them. So the sarcomere itself now is going to be the smallest contractile unit. It's the actual functional unit, you can say, of the muscle fiber, because now everything that's going to happen, everything that's going to take place within that sarcomere is going to be in relation to the contraction, right? The thick filaments are going to bind to the thin filaments, and then they'll pull and detach and attach and pull and detach again and reattach, and that'll continuously happen until you have full-on contraction. So everything that's taking place in one sarcomere is basically that same thing that's taking place in the next sarcomere, and then the next sarcomere, and the next sarcomere. So you get the idea, basically, what's happening there, as long as you understand what's happening in one sarcomere. So that's what we're going to break it down to. So now we're going to see, also we're going to see these sarcomeres, they're going to be aligning along myofibrils like boxcars of a train, and you can see that from the organization in this picture there as well. Now, here we're going to see, so the sarcomeres are going to contain A bands with half, basically, and I band at each end there as well. All right, so here we could see now, when we go through and we talk striations, we've got repeating series of dark and light bands. Now, those dark and light bands you can see in full-on detail here now. So we said, then again, contains A band with half I band at each end. The sarcomere, we're going to go through, we're going to be able to appreciate that. So here you can see that. Now, when we talk sarcomere, it's the region between two Z discs. So from one Z disc to the next Z disc, so the region between two consecutive Z discs. So here you can see you've got A band, and then you've got a little bit of a I band kind of right in there as well. And you can see it's composed of thick and thin myofilaments, and they themselves are going to be made up of contractile proteins. Okay, so they themselves are going to be made up of contractile proteins. So contains the A band with half I band at each end. You can see there as well. Excuse me. So the rest of that I band you can see is going to be contributing to then the next sarcomere you can see there on that side. So here then we look at one sarcomere. Now let's look at this in full-on detail. Here you can appreciate the A bands now are going to be the dark bands, the I bands are going to be the light bands. Now along with the A bands, the I bands give the cell as a whole its striated appearance. Okay, so when we talk A bands, along with those I bands, they give the cell as a whole its striated appearance. Next in here we've got the H zone you can appreciate. Now when we talk H zone, the H zone I want you to know is going to be found in the midsection or the lighter region of the A band. When we talk H zone is found in the midsection or the lighter region of the A band. So the A band you can see here running from this side here, okay, all the way to this side here. Now at the mid region of that A band, you've got the H zone. It's like the construction zone from here to here. So the H zone is from here to here, basically is what they're saying there. So you can see that H zone in there very nicely. Now the H zone we can see is important because it's going to contain this M line running right through it. It contains the M line. The M line, you probably recall from anatomy, is going to be a line of protein called myomycin. It's protein called myomycin. It's a dark line, and it bisects, you can see vertically, the H zone. It's going to bisect vertically the H zone. Then we can appreciate the Z discs. So we move to the I band. We can appreciate the Z disc, and we'll check those out there. Now, when we talk Z discs, they're proteins as well. These are proteins that are going to anchor the thin filaments. So you can see here, they are anchoring the thin filaments in place, while the M line did exactly that for the thick filaments. So the thick filaments are being held in place thanks to the M line. So the Z disc is holding then the thin filaments. You can see there in place. Next in here, we're going to be able to appreciate the term sarcomere. So sarcomere, I told you guys, is a functional unit of a muscle fiber. It is the functional unit of a muscle fiber, and it's the region between two consecutive Z discs. It will be the region between two consecutive Z discs. Myofilaments, then we've got next. Myofilaments are filaments that are going to be made up of actin, myosin, and other filaments. So when we talk myofilaments, they are filaments made up of actin, myosin, and other filaments. Now, when we talk myofilaments, let's look at the thick filaments and the thin filaments in greater detail. So here you can actually appreciate those thick and thin filaments in greater detail. The Z discs you can see there, the M line at the H zone you can see there as well. Now, when we talk thin filaments and thick filaments, I want you guys to know now thick filaments and thin filaments. The thick filaments are going to be our myosin myofilaments, and our thin filaments are going to be our actin myofilaments. Now, when we go through and we talk thick filaments, the thick filaments are composed of the protein myosin. So thick filaments composed of the protein myosin. And they're going to be found extending the entire length of that A band. So moving back here, you can see the thick filaments are found extending the entire length of the A band. They are found extending the entire length of the A band. Thin filaments, the thin filaments you can see there are composed of the protein actin. So you can see that there again. They're composed of the protein actin, and they extend across the I band. They are found extending across the I band and partway into the A band. And partway into the A band. We can also appreciate some elastic filaments here as well. We'll mention those in a few minutes. We'll check those out. Let's look at then the molecular composition of the myofilaments. Now, when we look at the molecular composition of the myofilaments, here we're going to be able to see now the muscle contraction is going to depend on myosin and actin containing myofilaments. So when we look at myosin, here we can appreciate the myosin then in greater detail. So here you can see the thick and the thin filaments. You've got the myosin and you've got the actin myofilaments there. Let's look at each of them in greater detail. So when we look at each of them in greater detail, now you're going to see the thick filaments. It's going through and it's telling you basically all of this information in regards to those thick filaments. I want you to go through and get that down there as well. We're going to look at the tails and we're going to look at the heads, and we'll go through those different parts there, and then we'll see how they're going to form cross bridges there as well. And then we're going to look at the ultrastructure of the thin filaments there as well, and we'll look at that full-on detail there too. So here's where it's going to take us, where we can appreciate the thick filaments and then the thin filaments. But again, we'll look at this picture. Again, it's giving us a more detailed image of what we're trying to look at, detailed view of what we're trying to look at. And then here you can see the thick filament in full-on detail by itself. So now when we go through, we talk about these myosin. Myosin is going to consist of myosin will consist of two globular heads. Two globular heads you can appreciate right inside of here. And two tails. Which are going to be intertwined. And we have a flexible hinge region between the two, basically. And we have a flexible hinge region between the two. And that flexible hinge region is just like what its name says. It's a flexible hinge region. This area is where you're going to see this this myosin is going to be able to kind of move into a resting position and then move into a cocked position where it's ready to form cross bridges, and then it could go back into a resting position. So that's all going to be happening due to the flexible hinge region we can appreciate there. So now when we talk the globular heads are the business ends. They are the business ends. You can think of the myosin. And the reason why we refer to them as the business ends is because during contraction, they will link the thick and the thin filaments together. They'll help link the thick and the thin filaments together, forming for us cross bridges, like we saw here. Forming for us cross bridges. Cross bridges are going to be formed during contraction. They are formed during contraction, and it's basically when the thick and the thin filaments are linked. When the thick and the thin filaments are linked. Now here we can see when we talk myosin myosin heads, you can see we also have here ATP binding sites. We also have ATP binding sites. Now, when we talk ATP binding sites, these ATP binding sites are going to be important because we're going to see here is where ATP will come and bind to. Now, when we go through and we talk contraction, we're going to see ATP is going to be required. Now, ATP is not only going to be required for the actual contraction, but ATP is also going to be required for the actual detachment there as well. So you have to remember that it's going to be required for both of those processes. And we'll talk more about that as we progress. Now, when we go through and we talk then actin, so let's move to the next slide. Here we can appreciate our thin filaments. Now, our thin filaments are going to consist of 2 strands of actin subunits. They consist of 2 strands of actin subunits. So actin is made up of protein. Protein makes up the thin filaments. Now, again, the actin, the thin filaments, are consisting, you can see, of 2 strands of actin subunits, and those 2 strands of actin subunits are twisted into a helix. And now you can also appreciate 2 types of regulatory proteins there as well, troponin and tropomyosin. I'm 100% sure if you work in a hospital, you should have heard of troponin, or you should have at least be familiar with that term there as well. So we'll look at that and we'll see how that's going to come into play also. Now, when we go through and we talk then actin, actin is made up basically of globular actin. So here you can see what we have is we have filamentous actin, or F-actin. This F-actin we see here is made up of many globular G-actin subunits. And that's what you're seeing, a whole bunch of these globular G-actin subunits down here. These G-actin, globular actin, are kidney-shaped polypeptide units. They are kidney-shaped polypeptide units. And you can see they bear the active sites. They bear the active sites to which the myosin is going to come and bind to, just like the myosin heads had the actin binding sites. So the globular actin units will have, you can see here, they bear the binding or the active sites to which myosin heads are going to attach to or bind to during contraction. So F-actin, when we get the G-actin subunits, and they get polymerized into long actin filaments, we form then what we call filamentous actin, or F-actin. So tropomyosin we can see here as well. Tropomyosin is going to be a rod-shaped regulatory protein. It is a rod-shaped regulatory protein. You can see it spirals around the actin core. It's found spiraling about the actin core. It helps to stiffen and stabilize the actin. It helps to stiffen and stabilize the actin. Now, in a relaxed muscle, like we're seeing here, in a relaxed muscle fiber, tropomyosin blocks myosin binding sites on actin. Tropomyosin, again, a relaxed muscle fiber, tropomyosin blocks myosin binding sites on actin. You can see that there. So in order for us to have cross-bridge formation occur, we have to move that tropomyosin out of the way. That makes sense, right? It should have made sense in anatomy. That's where you kind of saw the different anatomical components of this and where that came into play. But a lot of you guys come to this class from anatomy not even knowing what troponin is. Okay, so yeah, it's a very important component there, especially when you get into the physiology and the pathology of diagnosing diseases in patients. Myocardial infarction, most important one we've got there. So troponin then we can see there now as well. Troponin is another protein in relation to the thin filaments. Now when we talk troponin, troponin is a globular, it's a globular 3 polypeptide complex. It's a globular 3 polypeptide complex. One of its polypeptides is known as TNI. It's an inhibitory subunit known as TNI, and it's written, we can see here, as a capital T, lowercase n, and then we've got our I. So this is going to be an inhibitory subunit, and it binds to actin. Another is going to be, we'll see, TNT. TNT binds to tropomyosin. It binds to tropomyosin and helps to position it on actin. And it helps to position it on actin. And the third we'll see is going to be TNC. TNC. C for, you guessed it. I know you all got it right. Calcium. Because we're going to need calcium here as well, right? We're going to see calcium is going to come into play as well. So globular 3 polypeptide complex. You can see basically they're trying to depict that there in its structure as well. All right, so let's move forward then. So here now, as we progress forward, let's talk some elastic filaments. Now, when we talk elastic filaments, so here we're able to appreciate the thick and the thin filaments in this view here as well. But again, you can see all of those thick filaments basically with the myosin heads kind of coming right off and attaching to the thin filaments you have running in between. So thin filaments, you can see just by even looking at the structure, you can see thin and thick as you progress through. So here let's talk then some elastic filaments as well. So when we talk elastic filaments, they're going to be composed of basically protein titin. We talk titin. Titin is a giant protein. It extends from the Z disc to the thick filaments. And then runs within the thick filaments to attach to the M line. So when we talk titin is a giant protein. It's the first elastic, it's going to be this elastic filament I want you to know about. And it extends from the Z disc to the thick filament. And then it runs within the thick filament. And then it runs within the thick filament to attach to the M line. So again, you can see it holds the thick filaments in place. And it's going to help recoiling after stretch. And it's going to resist excessive stretching there as well. Next is dystrophin. Next protein I want you to know about here is going to be dystrophin. Dystrophin, we're going to see, is a protein and it links the thin filaments. It's going to link the thin filaments to the integral proteins of the sarcolemma. So it links the thin filaments to the integral proteins of the sarcolemma to help keep muscles intact. And also to help maintain alignment of the muscle. To maintain alignment and keep the muscle intact. There's a disease here I want you to know about, and it's called Duchenne's muscular dystrophy, or DMD, where these patients have no dystrophin. So without the dystrophin there, the muscles aren't able to exhibit that same integrity as a patient without this condition would be able to exhibit. So I want you to go through and get a little bit of an idea of Duchenne's muscular dystrophy there as well. You'll see these patients have hypertrophied calf muscles, okay, because these muscles now are being used excessively because they're not able to put, you'll see pressure on basically the rest of the muscles, and also the way they stand up is a characteristic maneuver they use to be able to stand up. Gower's maneuver, I believe, is what they refer to that as there as well. Next on, let's talk nebulin. Nebulin is going to work with other proteins that are going to act, basically, these other proteins are going to be myomycin and C proteins. And these other proteins are going to act to bind filaments or sarcomeres together and also help maintain their alignment. So maintaining alignment, keeping everything in place, helping with, you can see that muscle. So maintaining alignment. Next thing we said this was going to be then a highly specialized structure we're going to find in relation to our skeletal muscle. Right, so myofibrils we've taken care of there. One highly specialized structure there. Next is the sarcoplasmic reticulum, and then the T-tubules. So that's what we're going to look at here now. So here we can see now when we talk sarcoplasmic reticulum is a network of smooth endoplasmic reticulum. You know what smooth ER does. Smooth endoplasmic reticulum here is going to be special. Number one, you'll see because it's going to be found surrounding each myofibril like this, your sleeve does around your arm. So you can see that, I think it should be here, you can see how that sarcoplasmic reticulum is going to be found surrounding each myofibril, again, like your sleeve does around your arm. So the sarcoplasmic reticulum is an elaborate, smooth endoplasmic reticulum. It's an elaborate, smooth endoplasmic reticulum. It surrounds each myofibril the way a sleeve surrounds your arm, like I mentioned. Now, the major role of the sarcoplasmic reticulum is to regulate intracellular levels. Its main function is to regulate intracellular levels of ionic calcium. To regulate intracellular levels of ionic calcium. It stores calcium and it releases this calcium on demand. It stores calcium and it releases calcium on demand when the muscle fiber is stimulated to contract. Calcium is going to provide the final go signal for contraction. Calcium provides the final go signal you can think of. Go, like ready, set, go. So it provides the final go signal for contraction. It's basically the last thing kind of it's waiting for. ATP is there and calcium comes in and binds and boom, helps to expose the sites and contraction is going to take place. Then we have here also the terminal cisternae of the sarcoplasmic reticulum. When we talk about the terminal cisternae are going to be the enlarged, you can see here, look at these, here you've got all the sarcoplasmic reticulum. So here you've got the enlarged end sacs. The enlarged end sacs of the sarcoplasmic reticulum are going to be referred to as the terminal cisternae. Okay, terminal, end, cisternae, cisterns, basically storage units you can think of there. And what's inside the storage units? Calcium, as we mentioned. Then you can see now here in between two terminal cisternae, okay, between two terminal cisternae, you've got a T-tubule running through. I'm sure you've covered this in anatomy as well. Now the T-tubule is basically the invagination of the sarcolemma. It is the invagination of the sarcolemma. And the reason why this T-tubule is going to be important is because it's going to help transmit electrical impulses from the outside, right, to the inside. That's the only way we're going to be able to allow the inside to know, hey, there's an action potential passing through, is because the sarcolemma dips, it invaginates into the inside of the cell. When it dips and it invaginates into the inside of the cell, right, it's that T-tubule now that gets formed. So the action potential as it's passing down the sarcolemma dips right into the T-tubule. And the reason why it's going to dip into the T-tubule is because we're going to see it's going to excite the two structures that are right next to it, which are the terminal cisternae. Why do we need to excite them? Because we just said a few minutes ago, they're going to provide the final go signal, the calcium. So here now we can see when we go through and we talk the T-tubules. So the T-tubules are going to help transmit electrical impulses from the outside of the cell to the inside of the cell. They will help transmit signals from the outside of the cell to the inside of the cell. Now, what we're going to do is we're going to consider all three of those structures together. Now, when we consider all three of those structures together, we refer to them then as a triad. As a triad. Now, here we've got The relationship, now we talk of the triad, basically, is that you'll see the T-tubules and the sarcoplasmic reticulum are going to be involved in providing signals for contraction. The T-tubules are going to contain integral proteins. The T-tubules contain integral proteins. These integral proteins that the T-tubules contain are going to act as voltage sensors. They will act as voltage sensors. So when they sense that voltage change, obviously they're going to open up. And when these proteins then open up, they're going to excite the next proteins, which are going to be the proteins in relation to the walls of the sarcoplasmic reticulum. So those proteins of the sarcoplasmic reticulum are going to be called foot proteins. Foot proteins. And they form gated channels. They form gated channels through which the calcium can be then released. I told you they're going to be storing calcium. Well, yeah, we got that there, but then how are they going to release it? That's the main part. And that's what we've just mentioned there. So you're responsible now for knowing the names of these receptors. Okay, both the voltage sensitive for the T-tubules and also the receptors in relation to the sarcoplasmic reticulum. Okay, ryanodine is one of them. Okay, and I want you to look up the other one. D something something. Okay, D something P. I'll even give you more of a hint. Okay, you just have to look for one letter now. All right, so then when we go through and we talk then the sliding filament model of contraction now, when we talk about the sliding filament model of contraction, now here we're talking contraction. Now when we talk contraction, I want you to think of, when we talk contraction, I want you to think of then basically shortening. Okay, so we talk shortening of the muscle fiber. Now in physiology, okay, in physiology the term refers to the activation of our myosin cross bridges, which are going to be the force generating sites. Contraction is going to end. Contraction ends when the cross bridges become inactive and the tension declines. So now when we look at the actual sliding filament model of contraction, here we'll see basically the muscle in two different states. You can see the muscle in the top picture in a fully relaxed state where you have very minimal overlapping of the actin and myosin filaments. And here you can see how the Z discs are placed apart. The H zone you can see is clear. It's clear of any thick and thin filaments. Right? And the I bands are also spaced apart. But now when we talk in a contracted state, now what's going to happen in a contracted state is our myosin heads, they're going to get excited. They'll go and they'll attach to the thin filaments, and then they'll go and they'll pull the thin filaments in, and then they'll relax, and then they'll go back and attach again, and pull again, detach, reattach, pull, until you can see we have a full-on contraction, which we can appreciate then in this bottom picture. Now in this bottom picture, you can see that H zone has completely disappeared. The H zone is fully occupied now by those thick and thin filaments, where all we had were basically just the thick filaments and not even the heads. You had just basically the tails all right in there. And now here you can see the same thing has happened there, but the thin filaments now are occupying. Here now what's happened, as we said, you have this continual attachment, pulling, and then detachment. So reattachment, pulling, detachment. So when that continuously occurs, it's kind of like a ratchet. When that continuously occurs, leads us to the fully contracted muscle, as we can see in this bottom picture. Now, how does all of this occur? Here you can see that from these previous images. here we can see first we've got our nerve. The nerve is going to excite this muscle by releasing a neurotransmitter. And as you have learned, the neurotransmitter here is acetylcholine. So when acetylcholine gets released, acetylcholine is going to help generate action potentials along the sarcolemma of the muscle. Now, when the sarcolemma generates this action potential, the action potential then is going to be propagated. It gets propagated in all directions, right? And not like in the neuron it just went in this one direction. So here it gets propagated in all directions, and as it's being propagated along the sarcolemma, it dips right into the T-tubule. Dipping into the T-tubule, helping to release calcium. And you saw calcium. When calcium gets released, calcium, you told me, or you should be able to tell me, is going to go in and it's going to bind to? Very good. Troponin, just like you said. When it binds to troponin, you saw then troponin, okay, once it gets the calcium, troponin then is going to be able to move that tropomyosin out of the way. Now, when the tropomyosin is moved out of the way, it's exposing, right, the active sites for myosin on the actin. And we can then allow cross-bridging to take place. So here you can see you've got your voltage-sensitive receptors. I told you you have to know the names for. And then you've got your other receptors. You have to know the names for as well on the walls of the sarcoplasmic reticulum. So one activates the other, helping it to basically stop impeding the release of calcium. Calcium gets released. Calcium is going to make its way over to, then you can see here in greater detail, calcium gets released, and it's going to flood the cell, basically. And when it floods the cell, also inside the cell are those myofibrils which contain that troponin. So when that troponin then receives that calcium, that troponin is able to move the tropomyosin off of the binding sites on the G-actin, the F-actin, exposing those, allowing then for cross-bridge formation or contraction to occur. So again, then in the fully contracted state, you can see the H zone disappears, the I bands will come closer together, and the Z discs come closer together. They say his, H, I, and Z, his disappear and shrink, kind of. The H zone disappears, the Z and the I bands come together. They help this muscle to shrink. All right, so that's where we'll conclude then this first part.